Welcome back to the channel Next Generation Institute. Our motto, turning dreams into reality. So in your biology, the first and foremost chapter that you have is chromosomes in class 10. So let's start our class. First, what is a chromosome? Chromosomes are highly coiled ribbon-like structures that are formed by the condensation of chromatin fibers during cell division. Okay, I repeat, chromosomes are highly coiled ribbon-like structures formed by the condensation of chromatin fibers during cell division. So, chromosomes are highly coiled ribbon-like structures. What does it mean? Is highly coiled. Highly coiled means in this manner. This highly coiled, and it's a ribbon-like structure. Ribbon-like structure is the keyword. Highly coiled is the keyword. Condensation is the keyword. Please underline. Okay. So, ribbon-like. So you have seen a ribbon or you have seen a thread. THREAD, thread. You can also write thread-like structure. These are formed when the chromatin fibers get condensed during the cell division. What is cell division? We will discuss it in the later in this chapter. So, chromosomes are highly coiled ribbon-like structure. This is the diagram of a chromosome. Okay, this is the centromere. This is the sister chromatids. You have to label centromere, sister chromatids, and nothing else. Okay, it may come. Now chromatin fibers. So we know that chromosomes are formed when chromatin fibers get condensed. So now these chromatin fibers then consist of two things. One is DNA. We know about DNA, or we have heard about DNA, and it contains some proteins known as histone proteins. DNA full form is deoxyribonucleic acid. It will come in your exam for 1 mark at least, that what is the full form for DNA. And remember, you have to write DNA in full block letters. DNA, not in short form. DNA, DNA in full letters. Okay, next that comes is the molecular structure of DNA. So the shape of DNA, or a DNA molecule, was first studied by Rosalind Franklin in 1953. So question may come, who was the first person or the first scientist who studied about the DNA shape? And it was Rosalind Franklin in 1953. But the structure, the double helical structure of the DNA was finally worked out, was discovered by Watson and Crick in 1953. For this they were awarded Nobel Prize in 1962. Okay, Watson and Crick, both are different scientists, together discovered the double helical structure of the DNA. Again, the DNA is composed of some molecules. Those molecules are very large in size, so they are called macromolecules. It may come, what is a macromolecule? Again, DNA has or is composed of nucleotides. Again, these nucleotides are made up of three main substances, that are phosphate, sugar or pentose sugar. Both are same. You can call it pentose sugar also, or only sugar, and nitrogenous bases. Now here I have given you the diagram of the DNA. This one is called a double helical structure. This one is called double helical structure. You can see in this image, this is called the double helical structure of DNA, and this was finally worked out by Watson and Crick in 1958. Now what we got, DNA is composed or is formed from chromatin fibers. Chromatin fibers consist of DNA and histone. That DNA is composed of nucleotides. Nucleotides is again divided into three. There are three types of nucleotides, or there are substances that are composed of nucleotides are phosphate, sugar, and nitrogenous base. So now that nitrogenous bases is again composed of three things. Sorry, four things. Very sorry, four. There are four types of nitrogenous bases. Okay, one is adenine and guanine. In this picture also you can see I have mentioned adenine and guanine. I have highlighted it. This diagram is very important. It will come in your examination. These both form pairs. These four form pairs with each other. Let's take an example of a couple. There are a couple. There is a boy and a girl. They both love each other and they married each other in their later life. Okay, so there was strong bonding. There was strong bond with each other. Just like that, adenine pairs with thymine. There is a very good bond with each other. They are just like couples, and they have two hydrogen bonds. I have mentioned in and underline it with red color. And then guanine and cytosine, they are also they are very strong bonds, and so they are three hydrogen bonds. Now, histone proteins that help in the coiling and packaging of DNA into structural units called nucleosomes. The DNA strand winds around 8 histone proteins called histone octamer. Each such complex is called nucleosomes. So there are proteins. This is a DNA molecule. The DNA molecule, then the histone protein has the coiling. They coils each other. You can see the diagram. It is there in your book also. It helps in the coiling and packaging of the DNA into structural units. This is the DNA. The histone proteins helps it to pack the DNA, packaging and coiling of the DNA, and form it into the chromosome. After packaging, it forms a chromosome. Okay, sorry for the diagram drawn, it's not so good, I know. So it helps in the packaging and coiling and packaging are the keywords, and they are called nucleosomes. Note, a single human chromosome may have about 1,000,000 nucleosomes. It is not so important, but question might come in MCQs. Okay, chromatid. Now listen to it very carefully. Chromatid is one of the two identical halves of a duplicated chromosome connected by a centromere. Let us go to the first page. Here you can see this is a diagram of a chromosome. So it's a vertical half. So I draw a vertical half. Now it forms two blocks. These two and these two are called chromatids. This one and this one chromatid. This one also is chromatid. This one is also a chromatid. And these chromatids are joined by a centromere. The centromere. This is called the centromere. See, It is also written. I have drawn a vertical hub as per the definition of a chromatid, and it forms two blocks, two opposite blocks, and then these two hands, this one and this one and this one and this one, are chromatids. So let's go through the definition again. Chromatid is one of the two identical halves of a duplicated chromosome. We must mention duplicated chromosome connected by a centromere. Definition will come. Sister chromatid. Sister chromatids are two identical chromatids formed by the replication of a single chromosome held together at the centromere until they are separated during cell division. Let's go back again. This two, see here it is written. These two and these two are sister chromatids. This one and this one, see it is written over here. They are the sister chromatids. These two opposite telomeres. You can see the telomere it is written. These two opposite are sister chromatids. Okay, these two opposite hands. So sister chromatids are two identical chromatids formed by the replication of a single chromosome held together at the centromere until they are separated during a cell division. Definition will come. Now the centromere, this is the centromere. Okay, the function of the centromere is to hold the sister chromatids. They are the sister chromatids and serve as attachment for the spindle fibers. What are spindle fibers? We will discuss in the later in the chapter. During cell division, ensuring proper chromosome segregation, it makes the two sister chromatids to join each other, to be in contact. See, these two and this one are in contact. They are in contact because of this centromere. You see the red color. This one, this is the centromere. Okay. Now, genes, very important, very important. What are genes? Answer: specific sequences of nucleotides on a chromosome that encode particular proteins are expressed in the form of some particular feature of a body. They are the units of heredity. You will see your parents say, or especially relatives or your neighbors say that you look like a parent or your guardian. Or during, suppose your mother says that your eyes, your nose are just like your father or like your mother. Okay, the behavior you do, your behavior and your father behavior, when your mother gets very angry, she says that you are just like your father. Your father also does the same. Why she says that? There is a logic in there, because that is for the genes, this particular word, and that is the reason why they have written they are the units of heredity. Why they are the units of heredity? Because the characteristic feature that your parents have, that feature will get transferred to their offspring also, who is their offspring? You are their offspring. Your parents' offspring are you. So you will have some similarity with your parents, and that is for these genes. Sorry. Need for new cells. Why do we need? Just you need to know the names. There are big explanation in your book given, not required at all, children. Need for new cells. Why we need a new cell? For growth, for replacement, for repair, for reproduction. Suppose in your school there is a physics teacher or a biology teacher, but there is only one physics teacher in your school, and there are total 4 sections in each classes. It is very difficult for one physics teacher, or also for 2 physics teachers also, to handle 4 sections. So we need a new teacher. Why? For repairing, for replacement, so it can replace. For reproduction, we need a new teacher so that the burden in those 2 teachers on that one teacher is reduced. The same is here. New cells for growth. If that one physics teacher is not good, but he or she is not teaching physics very well, he or she can be replaced by another teacher. Okay, because he or she is not capable of teaching. Cell cycle. What is a cell cycle? Very important. Definition may come or may not come. Okay, not the definition. I don't think it is so. It will come, but then also you must study it. Cell cycle. Okay, cell cycle. The cell cycle is a sequence of events that a cell goes through to grow, replicate its DNA, and divide into daughters. Now, this cell cycle, and again, they are divided into 2 phases: interphase and M phase, or mitosis. Meiosis is not in your syllabus in children. Please make it sure and mark my words. It is not there in your syllabus. Meiosis is not there at all. Interphase, one of the phase of mitosis. Okay, the definition is not required. If you want to learn, you will learn. First growth phase, second growth phase, synthesis phase. Okay, first growth phase, they are the the interphase consists of G1, G2, S. G1 means first growth phase, G2 means second growth phase, S means synthesis phase. Okay, now this first growth phase, what happens in the first growth phase? The RNA, not the DNA. Mark my words, listen it very carefully, children over here. RNA and protein synthesize. They are synthesized mitochondria and chloroplast. They divide. In synthesis phase, the same thing happens. RNA and more synthesized, but in a greater manner. More RNA and proteins are synthesized. Synthesis phase. DNA is synthesized, not RNA. Don't get confused with it, children. DNA is synthesized. Chromosomes duplicate but remain attached. Here is a diagram of a cell cycle. See, the diagram is also given in your book. Okay, now this diagram, replication of DNA is important. This one is very important. It may come for drawing. Okay, important question. For 1 marks or 2 marks it will come. Can the cell cycle go on endlessly? No. At some places, sorry, it is dome. Oh my god, I am very sorry. It is some. It is some. At some places it stops permanently. At some places it stops temporarily. Cell division. Okay, now cell division. This cell division is divided into two phases: mitosis and meiosis. Meiosis is not in your syllabus. Mark my words. But what is meiosis and its significance? You should know. A picture of the cell division I have taken from a book. Mitosis is the cell division leading the production of diploid. Diploid means two cells or two chromosomes, two diploid for growth and development. See here, interphase, prophase. You can see the difference. Central starts moving. Chromosomes become distinct. You can see the chromosomes very clearly. Here is the chromosome I am drawing. Here is the chromosome. You can see it very clearly. Only early prophase is required. Late prophase is not required. This is from your book. Now, metaphase. Chromosomes line up. You can see the chromosome has lined up in a single line, a single vertical line. Each chromosomes get attached to spindle fibers by its centromere. What are spindle fibers? You have the spindle fibers. See, there are some lines you can see like this. Where the chromosomes are lying. These are called spindle fibers. Here the chromosomes are lying, and you can see some lines like this. These are called spindle fibers. Plant cell, animal cell. Remember, animal cell is in circular. It is because it is no cell wall, and it has a cell wall. Plant cell wall. Okay. Now, anaphase. See, this is also required. This one is also required. Okay. Anaphase. Centromere attaching the two chromatids divides or splits. It gets splits. You can see the chromosome divided like in this manner. Like this. Lesser than, greater than. Like this. In this way they have been divided. In anaphase, the chromosomes gets divided into opposite poles. Okay. The same thing happens in anaphase. The two sister chromatids of each chromosome separate and are drawn apart. They are apart towards opposite poles. In opposite poles, pulled by shortening of spindle fiber. So these 2 points are important. Now, in late, see, students are confused over here. In late anaphase, what happens is that there is a little bit of furrow. There is a furrow, little. If this diagram comes, you have to see the difference. Wait a minute. In telophase, see, there is a deep furrow or deep gap, but here only little. small. So you have to understand difference. If this diagram, if late anaphase comes, you have to be very conscious. You will write I fear and I'm afraid, or afraid that you will write telophase. Don't write see the furrow, a deep furrow, but yeah, not so deep. So I have told what is the telophase. This is the main point. Furrow appears. Two sets of daughter chromosome reach opposite. Now these are daughter chromosomes. This one and this one. These two. This one is a daughter chromosome. Spindle fibers disappear. Now here spindle fiber disappears. The spindle fiber we have seen like in this position. In this way they disappear. In plant cell also. Now nuclear membrane is formed. Cleavage furrow starts deepening. This is a deep furrow. There is a deep. This is a deep furrow. Nuclei reappear. The nuclei also reappears. Cytokinesis, very very important. Cleavage furrow deepens. It gets so deep that it divides two daughter cells. That again interface happens, and the two daughter cells get divided. But here there is a plate, a cell plate. You can see here also a little bit of cell plate in telophase. You can see they don't get divided like this because they have a hardy shell wall. And that's why they are divided by a cell plate. Now we will study the what is the difference between animal mitosis and plant mitosis. Definition, you know the definition of animal mitosis means the cell division process occurring in animal cells in order to increase the cell number and repair damaging cells. In plant mitosis, the cell division process occurring in plant meristem tissues in order to grow. Okay, see difference may come for 2 marks. 2 to 3 marks, or for 1 marks also it may come. Mark my words. Centrioles. In animal mitosis, you will see the centrioles are present during mitosis, but in plants you will not see the centrioles anywhere. Location. Animal cell occurs throughout the body. Throughout the body animal cells occurs, but plants only occurs in the meristematic tissue or in the meristem tissues. During cytokinesis, what happens is that they create a cleavage for the deep furrow, but the plant cells they have a cell plate. There grows a deep cell plate. I will show you the image again. See, there is a cell plate. This is again a cell plate. They don't get divided. Aster formation occurs in animal, does not occurs in plants. What are asters? I will show you what are asters. Wait a minute. This kind of thing you will see. These are called asters. This is aster. You will see here also. You can see the asters are mentioned. This one, but in plant cell you will never see. Significance of mitosis: growth, repair. Wait a minute. Okay. Growth, repair, and replacement. There is a growth or increase in the body size due to the formation of new cells, because new cells form in tissues. So there is a growth, repair. If there are some tissues which gets damaged or they get wounded, so they get repaired by the new cell, and the cells which get older or they are dead cells, they are of no use, they get replaced by the new cells. Okay. Now meiosis, we will a detailed analysis of meiosis. Meiosis is the kind of cell division that produces sex cells or gametes. Okay, I have told you the cell division of meiosis is not in the syllabus, but significance of meiosis you have to learn. Genetic variation. Meiosis introduces genetic diversity throughout crossing over. What is crossing over? Exchange of genetic material between homologous chromosomes and independent assortment. This variation is essential for evolution and adaptation. Very important point. Okay, mark my words. Formation of gametes, maintaining chromosome number. Meiosis didn't occur, chromosome numbers would double with this. There's a in each generation of meiosis, the chromosome number will get double, means 2x. Sorry, 2x. It will get doubled. So we have come to the end of the chapter. Then last 2 points. Elimination of mutation, evolutionary significance, but these are not so important, but then also I have mentioned you, but not so important. Okay, so here is the end of the chapter chromosomes. So we are done with the first chapter in your class 10 syllabus, that is chromosome. Chromosomes we have done. So at first, what we have to learn? Definition of chromosomes, diagram, what does chromatin fibers consist of, who was the first person to study about the DNA and a double helical structure, was first worked out by Watson and Crick. The year is not required. Macromolecules, DNA, nucleotides, the 3 components of nucleotides, diagram of DNA. There are 4 types of nitrogenous bases: 2 hydrogen bonds, adenine pairs with thiamine with 2 hydrogen bonds, guanine pairs with cytosine with 3 hydrogen bonds. Histone proteins, the definition: chromatid, which is a sister chromatid, which is a chromatid. You have to know what is the function of centromere: to hold the sister chromatids, to hold it, to attach the sister chromatids, to make them attached. That is the function of the centromere. You have to learn it by heart. What are genes? Definitions will come. Need for nuisance, only names. Children, only names. Explanation not required. Cell cycle definition not so important, but also you must learn. 2 phases. The interphase is composed of 3 phases: first growth, second and synthesis. We are going to a very short recap for the whole chapter. For RNA and protein synthesis, mitochondria and chloroplasts divide, very important. More RNA synthesized, very important. Synthesis phase, more DNA synthesis, chromosome duplicate but remain, that is very important. There is a diagram, it will not come for drawing, okay, but this one will definitely come. Diagram of DNA replication. Now I've made you understand all these things. The cell cycle, they can, at some places it stops permanently, at some places it stops temporarily. The cell division, what is meiosis, what is meiosis, the types of cell division, the cell division of meiosis, prophase, interphase. Now the interphase, prophase, and metaphase, anaphase, telophase, cytokinesis. The diagram are very important, children. Mark my words, diagram are very important. Diagram for mitosis is important. Okay, study the diagram, very hard. Study the diagram and practice diagrams. This will definitely come for 2 marks or for 1 marks. I have written over here. Or for 1 marks. Significance. Definition of meiosis, significance of meiosis, and here we come to an end. So this is a short recap. Okay, thank you students for watching, and my name is Shubhadeep. Shubhadeep sir. Okay, thank you for watching. Stay safe, stay fine. Okay, thank you very much. Next Generation Institute, our motto is turning dreams into reality. Thank you.